last time we met, we were talking about eukaryotes. We were talking about the different diseases they cause. Just as a recap, I mentioned to be careful with the terms Tinea and Tinea pedis. Are those genus and species?
No.
No. Excellent. They're not genus and species. They sound kind of like it, right? But what are they in reference to?
Location.
Location, where it's at. Excellent. Good job. So location and where they're located. So Tinea is on the head. Tinea pedis would be on our feet, like athlete's foot. We mentioned that the treatments were challenging. Why is it harder to treat these infections, these fungals, these yeast infections, than it is bacterial infections? They're similar to our own cells. Yes, exactly. So they're similar to our own cells. So we have to work a little harder to get rid of them. But we also have to be careful with the amount of antibiotic we use or the amount of antimicrobial growth, because even though these are microbial eukaryotes, they still are similar to us. Similar enzymes, similar processing. Okay, so that's where we pretty much left off. We mentioned that a lot of fungal pathogens are opportunistic. What does that term mean again? They have an opportunity, they'll take it. Good. So they're okay in some areas. If they move to a wrong area, then they're not okay. Another thing, too, is they're okay as long as they're in balance. I mentioned that a lot of times women are prone to yeast infections when they take antibiotics. We take away the normal flora of the vaginal region, and yeast, which is normally there at a ratio of 50-50 with the bacteria, can now take over, and our flora can become more along the lines of 100% yeast. So we have to take an anti-yeast treatment as well a lot of times when we're taking those antibiotics. Another thing, too, we mentioned we're worried about our compromised hosts. So people who have immune systems that are lowered or who are weakened. And the biggest thing, of course, we always worry about is lung infections because they are so hard to treat. This was our slide we left off on, which was our whole new phyla that we started to talk about. And so this is where we're going to start talking about our grouping that are more of the algae. So we just finished talking about all of our fungi and our yeast. And so now we're switching gears and we're going to talk about our phyla that are going to be algae. And there's a few organisms in here as well that can be pathogenic, which is why we're going to talk about these groups. So in this grouping, they go under the classification of the Protista. And the thing with the Protista is this is kind of like a junk drawer of biology. If we can't fit an organism perfectly into, the kingdom Plantae or to the kingdom Animaliae, we put them into the kingdom Protista. It means that they don't fit perfectly into any other category. So like I think I mentioned that before, it's like our little junk drawer that we all have at home, right? We're just not sure where they go, so we classify them here. Now, it doesn't make them any less important. They're still important, but they're just a little bit different from everybody else. So the idea behind this then is we're going to have them separated into 5 main phyla and 5 main divisions, and we're going to talk about these and how they relate to our microbes. So the first one are the brown algae group. This grouping is referred to as Phaeophyta, and these are a lot of times are separated into their 5 groupings based on what kind of chlorophyll they carry. Chlorophyll, remember, is important for photosynthesis. So their type of chlorophyll is chlorophyll A and C. They also have a pigment associated with them called xanthophyll, which can also be kind of like a yellowish brown pigment. Now, the reason I like to talk about these ones is because these are ones that are associated with our central coast. If you have ever gone diving or if you've ever gone off of the coastline, you see all of those large kelp beds, they would fall in this grouping. So a lot of people think that kelp are plants. They really aren't. They are really a type of algae. Now, of course, they're non-modal. As we know, they stay stationary. They have little anchors that hold them in place. But the reason they're important to us is inside of the kelp, we can harvest them for a type of protein they produce and a type of carb they produce, which is referred to as an algin. This is a thickening agent that we use in a lot of our shampoos, our conditioners, and our foods. So if you've ever read the back of like a food or a shampoo label, sometimes you'll see algin mentioned. That can be collected right along our coastline. That's pretty cool, right? So that's kind of a neat thing. Another thing is that they are also going to store their extra sugar as a type of a mannitol. And we've heard of mannitol before. What did we use mannitol in? Yeah, our agar, right? For our Staphylococcus aureus, remember we used mannitol salts, right? And so we were using that to differentiate Staph aureus and Staph epi. So these are a good mannitol source for us as well. The next group is probably the most important to the microbiologist. This is the Rhodophyta group. And these are known as the red algae. They naturally have a red pigmentation to them, partly because of the fact that they have what are called phycobilin proteins inside, which give them this really pretty red coloration or pink coloration. Now, they're also going to have cellulose cell walls, which means they're going to be fairly rigid. And the most important thing for us is that these are going to be our agar sources. So remember I told you that agar is something that bacteria can't eat or break down, unlike our old-fashioned friend gelatin that we used to try to use. And so because of that, we harvest a lot of our red algae to be able to use them in our agars. Chlorophyta. So chlorophyta is a typical green-colored algae. And this one is going to store its extra glucose as kind of like this polymer that it forms that is going to be used also as a basis in some of our foods and also as a basis in some of our agar types to grow our bacteria. We believe that the chlorophyta were early ancestors for our plants. They're very similar to plants as far as producing the amount of photosynthesis processes they use, the type of photosynthesis they use, and also the fact that they have that natural green coloration that we see with our plants. If we look at their cells, they also have a lot of other biochemistry that's similar to what we see in our plant cells. You probably see them most often if you go to Oso Flaco or if you go to like a pond as kind of that greenish pond scum that's growing on the surface, right? The kind of stuff we try to avoid when we're wading. If we go to Lopez Lake or something like that, that would be your chlorophytas. So your pretty green algae. Our diatoms. So we've mentioned diatoms in here when we used our diatomaceous earth, right? So I told you diatomaceous earth we purchase, it comes from Lompoc. There's a couple other areas, but Lompoc is one of our main distributors, which is just in our backyard. You can actually see where they harvest it. If you drive towards Lompoc, it's on the south end of town, kind of close to where the dump is. If you're familiar with that, in the hills, you'll see kind of like a little white cliff region, and that's where they actually harvest their diatomaceous earth. So what is diatomaceous earth? Well, it's actually a collection of dead diatoms. And diatoms are little microscopic organisms that are found in oceans and in marine water. So does that mean that Lompoc at one time was underwater? What do you guys think? Yeah, definitely. So there was a shallow sea there. Yeah. And we see that. We see the signs of that shallow sea being present. These are probably some of the most beautiful organisms that we ever look at under a microscope because they tend to have a silicate structure, which means they look like a glass. And so when we look at them, it's like looking at a shard of glass under the microscope. They catch the light and they're kind of rainbowy looking. And you can see in the picture here for my zoomers, this wagon wheel that I'm circling here, or over here for you guys in class. They make these really intricate patterns that are really beautiful. A lot of times they also tend to make like almost like a box and lid structure. So they have these kind of cool 3D structures where they'll attach to each other. And so they have these really beautiful formations. Now, the reason that we're interested in them is because they produce something called domoic acid. And domoic acid, we'll talk about it in a few more slides, is something that can affect our neurology. So we're going to talk about domoic acid in just a few more slides. The fifth group that we're going to mention are the dinoflagellates. And these dinoflagellates also tend to be unicellular, just like the dinatoms, but they are flagellated. That's why they're called the flagellates. And so that means that they are modal when they're swimming around in the water. These are what we think of and when we picture plankton. We hear about sea creatures, eating plankton or filter feeding on plankton. The dinoflagellates would be included in that plankton category. Now, the reason they're important to us is because these guys are known for the phenomenon of red tide. Have you ever heard of red tide before? Yeah, good. I've seen lots of nods. What are we supposed to avoid eating when there's a red tide occurrence?
Mussels.
What's that?
Mussels.
Mussels, yeah. Mussels, clams, oysters, any kind of a shellfish, like a bivalve shellfish. We have to avoid them. And the reason for that is clams, mussels, oysters, they filter feed. And so they're pulling in all of our dinoflagellates that are present and using them as a food source. Dinoflagellates sometimes will have natural blooms. And what that means is their numbers will increase a lot. And so maybe the environmental conditions are just right, temperatures are just right, and so their population just goes up astronomically. When this happens, that means that our clams, our mussels that are filter feeding are eating even more than normal. And unfortunately, as they start to eat more than normal, some of the components found inside of our dinoflagellates start to accumulate in their muscle mass or in their tissues. And so that's what we see when we see the advancement of red tide. So what's accumulating? Well, it's basically going to be a neurological toxin that our dinoflagellates can basically produce as they start to grow in the environment. Now, if I was to eat one dinoflagellate, I wouldn't get sick. But remember, they're filter feeding, so they're bringing in thousands, if not millions at a time. And so what happens is it accumulates into the tissue over time. So if I eat one of these clams or mussels, then I'm going to get exposed to this toxin. Now, this toxin is paralytic. And so what that means is it'll affect our nerves. And so if you eat a clam or a mussel during a red tide event, you will start to have tingling in your lips. You'll start to have tingling on your face, very similar to what we talked about when we were talking about how Clostridium botulinum can make your face start to go slack. Very similar to that, except this tingling sensation will keep moving and you'll start to have more and more paralysis and less control of your facial muscles. The reason we worry is because this paralysis will also expand to the trachea. It will also expand to the lungs. And so what this means then is you're not going to be able to breathe. And so it can be potentially deadly, depending on how much you consume. So this is why when there's a red tide event, we want to be careful with our consumption. Yeah.
If you don't like a sufficient amount for it to reach the lungs or anything like that, does it wear off?
So it doesn't usually wear off on its own. If you've eaten enough to where you're getting a tingling sensation, you're going to have to seek medical help. So if you're feeling those pins and needles and those tingles, definitely don't hesitate. Go in and see medical help. Yes?
So where would bioluminescent algae come in?
Good question. So our bioluminescents would be ones that they can be in different groupings, but there are some of the dinoflagellates and some of the diatoms that can do bioluminescence. And I'll actually show you a picture of red tide at night. And it's really pretty. It actually has like a fluorescence color to it. It's really neat. So with this picture, we can kind of see why it's called a red tide, right? It has kind of that reddish tone to it. But you can actually tell in regions on the Central Coast, and I'll show you a picture in a minute, that when we're having a red tide event, because you'll actually see like a little bit of a glow to the waves. And it's very subtle. It's almost like a moon glow when you see it. That picture on the far end is showing a whole bunch of dead fish. And the reason for that is because the fish are also feeding on other fish that are feeding on filters, meaning that some of their prey are filter feeding. And when the fish eat the prey that have been filter feeding, they too can get exposed to the neurotoxin. So the good thing, and we'll come back to that a little bit more in just a minute, the good thing about our algae is that they're responsible for a lot of photosynthesis. And what that means is they contribute to about 80% of our oxygen on Earth today. So our rainforests and our algae are huge. They work together to be our oxygen producers. If they're doing photosynthesis and they're producing oxygen, that also means that they're using and absorbing CO2. So they're also considered to be those carbon sinks, as we talked about, where our carbon drops a little bit. They tend to be the foundation of a lot of food chains. So they're going to support a lot of our larger organisms out there. And then we use them for a lot of our products. We use them for our agar. We use them for our diatomaceous earth. We use them for algin. So we have a lot of different uses that are very helpful for us. So lots of good. The bad is that, as we mentioned in those other slides, they can cause illness. So I wanted to bring this one up. We talked about the red tide with the dinoflagellates, and we said that one of the concerns we worry about is paralysis with those. Another concern we worry about is with our diatoms. So diatoms are also found in the water. They're also used by organisms that are filter feeding. And then those fish can eat those filter feeders and get exposed to domoic acid. And then sea life, like sea lions and seals, could eat those fish, and then they can have a buildup of domoic acid. And at Pismo Beach, about 3 years ago, we had a really odd incident, and you can see the circle here, where a young girl who was 14 was wading in the water and ended up having a sea lion come up and bite her leg on the calf. And thankfully, her parents were filming it because at first, when it was reported, people thought, oh, she must have been messing with the sea lion. There must have been something going on. But no, she wasn't. It actually just came up and bit her for no reason. It was just in the waves and got her. And it was like, only in about 14 inches of water. So, of course, 911 was called. Of course, the lifeguards go out to try to figure out what's going on. They find the sea lion and it's over on the sand and it's like snapping and biting and almost acting like it's rabid. So they don't know what's going on. So they unfortunately have to put it down. But they start looking in and trying to figure out what is wrong with the sea lion. And what they find when they do a brain scan is what we see in this top picture here. So at the top, we're seeing a normal hippocampus, which is a part of our brain that's going to control reasoning, movement, processing. And you can see what the sea lions look like, and that's a swollen hippocampus. So what that means then is that the sea lion was exposed to this high level of domoic acid. It caused internal brain swelling. And so it was no longer able to process things like normal. Again, it was almost like rabies, where it was losing control of its own neurological function. So this is just kind of a reminder that if these diatom levels get up, they can potentially be dangerous for us. So we have to monitor these. Same is true with our red tide. We have to monitor them to make sure that we don't run the risk of getting ill. Now, that gets us through our algae group. That's going to lead us to the next group in our proteustas, which are referred to as the protozoans. The protozoans are going to be ones that are more similar to animals. So algae are more similar to plants. Protozoans tend to be more similar to our animals. Now, to fit in this grouping, they are all unicellular. They're all chemoheterotrophs, again, just like us. They have a lot of different forms associated with them. So a lot of times when conditions are bad, they can form what's called a little cyst shape. Think of it as being like an endospore, okay, kind of like what bacteria do with those little spores. And then when conditions get good, they'll hatch out and start growing again. And so those can be referred to as trophozoite stages, meaning that they're growing and actively living in a new environment. They can reproduce sexually. Some can do asexual, meaning that they'll produce little clones of themselves. Most of them are aerobic, and most of them are going to require a high moisture environment or a host to be able to survive. They also are like animals in that they don't really have a true cell wall. They're going to have what's called a pellicle, which is basically like a protein carbohydrate covering, but it's not a true cell wall like what we see with our plants and with our algae. So again, that life cycle they go into is they can have a vegetative state, just like our bacteria, where they can grow and reproduce and use their nutrients in their environment. But when things get rough, they're not getting enough nutrients or they're stressed, they'll form a cyst stage. And again, think of that as being like an endospore. And then that cyst can basically stay in kind of this, suspended animation until it gets inside of a new host or inside of a new environment. Once it's in a new host or environment, the cyst will hatch out or open up, and we start to get our vegetative growth again. So they will constantly go back and forth between trophozoite and cyst stages. Now, because of this means that we have to be careful with food and water consumption. If we eat or drink water that has cysts in it, for example, then we run the risk of getting sick because that cyst can go into our intestine, hatch out, and become a trophozoite. We're going to see a picture of one of our most common ones on the next slide, and it's probably one of the most cutest ones too. Also, we have to be careful because a lot of these are also spread by organisms like insects or vectors, where they can stay in a cyst stage inside that insect or vector. until they get put into a human body, and then they can hatch out and become vegetative. So let's explore some of our protozoans. So one of the groups that's in here are the archaezoa. And these ones are going to not have any type of mitochondria, which means that they're not going to be able to produce their own ATP. So what that means then is that they are going to be reliant upon us or any type of host to provide resources for them. And as we've talked about before, if they can't produce a mass amount of ATP through using Krebs and UTC, then that means that they're going to take a lot of our nutrients. And so we're definitely going to feel when we have these kinds of infections. So one of the most common ones that we see in our area are ones like Giardia. There's a picture of Giardia on the bottom. I'll circle it here. And Giardia is one of the reasons why we can't drink the water at Lopez Lake directly. Or you can't go hiking and find a pretty stream and drink Giardia or drink the water directly, because you will be drinking Giardia. So where does Giardia come from? A lot of times it's transferred from fecal deposits by animals that are defecating or urinating in the stream, so it can come in through that way, which is not something we want to drink anyway, right? And then we drink that water and we can ingest cysts. And then these cysts can move into our intestine and they will hatch out to their vegetative trophozoites and they start to grow. Now, the reason I like Giardia is because they have the cutest little happy faces. Do you guys see the little happy faces? You can just kind of make them out. They have like these little eye spots and a smile. They have these little eye spots and smile. So when you're looking at a microscopic slide of somebody that has Giardia in a fecal sample, you actually see these little guys kind of smiling back at you. I mean, it's kind of fun. You feel bad for the patient, but it's kind of funny at the same time because they're like, hi, yep, we're here. So we can treat them. Luckily, there's treatment. But the problem is a lot of times patients don't even know they have this. They just know that they're having diarrhea, upset stomach, and it doesn't usually easily go away, which means that they do have to seek medical treatment. They'll know that something's wrong because they've had diarrhea for several weeks. And so that's usually when they reach out. And it's just a quick fecal check that we can do to look for them. Another one that's in this same group with the archaezoans are going to be one that's called Trichomonas vaginalis. And it's a common STI or STD. This is one that a lot of times will infect the urethra and it will cause inflammation. So again, you're going to know if you have it because you're going to have painful urination. Because the Giardia and the Trichomonas have multiple flagella, they tend to have an interesting movement. When you look at them on a slide, they tend to have kind of a ticking motion because their flagella are kind of all ticking in different directions or different ways, and it makes them kind of jerky. And so when you're watching them, if you see this kind of ticking motion, you know that you're looking at an archaezoan. We also in here can have our next group of pathogenic protozoans, which is one that refers to the apicomplexa group. And apicomplexa are going to be ones that tend to have pretty complex life cycles. And we won't go into them in detail here. Just know that, again, they can start as a cyst, and then they can be presented to the body, a lot of times either by a vector or by dirty food or water. And then once those cysts get in our body, they can hatch out and start to grow as a vegetative trophozoite. So similar process. Now, the one that we worry about the most is Plasmodium falciparum. You've probably heard of that as malaria. And remember, we talked about malaria with sickle cell in here, right? We looked at it before. So malaria, as we mentioned before, is carried by mosquitoes, and I'll show you a picture of its life cycle on the next slide. And again, you don't have to memorize it just to understand how it works. It's carried by mosquitoes. When mosquitoes take a blood meal, they inject us with some of the cysts. Once they get into our bodies, they hatch out and start to grow. And what cells did I mention that they tend to infect? Does anybody remember from sickle cell? What cells did they target in our bodies? Excellent. Red blood cells. Good. Yes. So what they're going to do is once they hatch out, they're going to find red blood cells, and they're going to go inside of the red blood cells and start to finish their life cycle. Once they're in the red blood cells, they're going to produce offspring, and then those offspring are going to hatch out. And when they hatch, they lyse or destroy the red blood cells. And they pop them open. So that's when we start to see things with malaria like the fever, hallucinations, and the potential for death if the fever gets high enough because we have so much loss of red blood cells all at once. They tend to go in cycles. Another one that we do worry about is an organism called Cryptosporidium. And we aren't as concerned as we used to be because we've learned how to screen for it. But Cryptosporidium was responsible for one of the biggest water outbreaks in history, and that occurred in Milwaukee in the 1993. And I know a lot of you weren't even alive at that time, right? But in 1993, in Milwaukee, we were not checking for this organism, for Cryptosporidium. It was usually associated with lakes and streams, and we were cleaning our drinking water. But somehow, in some way, we ended up getting Cryptosporidium in the city water supply. And it was something that we weren't checking for because we'd never had that happen. And so unfortunately, we didn't have any type of cleaner or preventative method in our drinking water at that time. We do now. We screen for it, and we also add an anti-parasitic to make sure that the organism doesn't grow if it is present. But at that time, we didn't. And so what happened was homes that were using their water from their faucets to cook or to drink directly ended up getting exposed to a high amount of cysts of Cryptosporidium. Now, for most people, that caused diarrhea, upset stomach that lasted about 4 or 5 days, and then they recovered. Unfortunately, though, for immunocompromised, for HIV patients, people that have reduced immune systems, it actually resulted in a lot of deaths. And they figured out that there were about 403,000 cases within 2 weeks. So that was huge. Again, we don't have to worry about it today. We do check for it today, which is good. So this was a learning experience, but unfortunately it involved a lot of people getting sick, and unfortunately a few people dying because of that learning experience. As promised, here is a picture of Plasmodium. So again, this is going to be our malaria organism. And again, you do not have to memorize it. I'm just sharing it with you so that you can kind of see what I was explaining. It starts with a bite of a mosquito. So at the top with our bite of our mosquito, what happens then is that's going to inject us with our oocysts, which are going to go inside of the body. So we can see that here in the middle. They're going to move to red blood cells, and they're going to start to do reproduction. Those red blood cells will eventually get more infection and eventually lice as their offspring hatch out. And unfortunately, then what can happen is if a mosquito comes and feeds on that person who has been infected, that mosquito gets infected and now can spread malaria to a new person. So that's why we worry about the cycles. Once you're infected, you can potentially infect others through mosquito bites. Now, an interesting thing about malaria. Have we ever had malaria here in the United States? What do you guys think? I see a few nods, a few nods. Okay, what do we think? Let's raise our hands. Who thinks we've had it before? And you guys can tell me yes in chat. Who thinks we've had it before? Don't be shy. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Okay, we got 12. And then who doesn't think we have? 1, 2, 3, 4. You can't vote twice. Oh, okay. You're like, I'm just not sure. I'm going to do both yes. Okay, so, all right. So we have 12 yays and 12 nays. So we're like right in the middle here. Well, I will tell you, we've had it before. As a matter of fact, have you guys ever heard of this company called the CDC? Yeah, the Center for Disease Control. Yes. Have you ever wondered why it was established in Atlanta, Georgia? Like, I mean, isn't that kind of a weird place to establish a huge mecca? Usually we see that in New York or LA, right? Well, the reason is because we had an outbreak of malaria in Atlanta, Georgia. They ended up having a shipping unit come in near Atlanta, and they had mosquitoes, unfortunately, that were in this shipping container. It wasn't like they were shipping mosquitoes. They just ended up getting in there. And so the mosquitoes came out, started biting people. They were establishing themselves as a good area for malaria because it's humid, it's moist. It was a great area for the type of mosquitoes that carry malaria. And so the government set up a small fraction, a small group of top scientists and researchers to get the problem under control. So they studied how malaria spread. They realized it was insect spread. They realized specifically it was mosquito spread. So they started isolating and trying to treat patients who had it to get their malaria counts down. Again, we can't get rid of it completely, but we can get their counts down. And they started doing a massive pesticide spray to get rid of mosquitoes in the area. And they were successful. They did an education program. They told people if you have standing water, dump it out. If you see mosquito larvae, dump bleach in the water. I mean, they worked together. It was a huge thing. And so they did such a good job, they eradicated the malaria. We still don't have it here today. We could if we got the right mosquito in, but luckily the type of mosquito we have here doesn't carry malaria. That's the one thing that's saving us right now. So the CDC did such a good job, the government decided to start giving them more funding and to let them work on other diseases. And that was the birth of the CDC. Isn't that cool? It's kind of a neat story, right? It all came about from malaria. Okay, one of the ones that wasn't on my previous list, but I love to mention it, is an old organism called toxoplasmosis. The reason I like to mention this one is there was a recent article that came out in 2020 that talked about the impact that humans have on toxoplasmosis. Thanks, Vishala, I see that. So, yeah, I see that. Thanks, Vishala. So I have a question in Zoom saying, are there numerous cases of malaria each year now? And so we now that we have it under control, there's not anymore, but there was during the outbreak, yes. So with toxoplasmosis, it's a little organism similar to malaria. It's spread through oocysts, and then it will hatch out inside of us. And is carried by a little, usually in a mouse or in a cat or in a farm animal. And so the vector process is usually that this little organism will get into a mouse, it will infect this mouse, and it will start to hatch out. And to complete its life cycle, it can't finish producing offspring in the mouse because of the biology that the mouse has. It needs to finish producing its life cycle inside of a cat. It's just because of the biology. And so what this little organism does is it actually takes over the mouse, kind of like what we saw in cordyceps. It starts to affect its brain, and it makes the mouse start doing risky behavior. Now, if you're a mouse, one of the riskiest things you can do is go up to a cat and go, nee, 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 nee. Well, guess what this mouse does? It does that. The mouse actually becomes attracted to cat urine, to the smell of it. So it starts stalking cats. It's like smelling where the urine is and trying to figure out where cats would be. And then when it sees a cat, it literally goes up on its little hind legs, or it rolls over, or it'll just kind of move around, and it'll wait for the cat to attack it because it wants the cat to consume it so that then the organism can finish its life cycle. Again, it's like invasion of the body snatchers, right? This thing is taking you over. Okay, so how do we get involved in this? Well, we get involved in this if we have an outdoor cat that has consumed a mouse with toxoplasmosis. And if we don't clean our litter boxes every single day, the toxoplasmosis will come out in the fecal matter of the cat. And then if we don't clean it every day, the fecal matter will dry out and little oocysts, little cysts, will be in the feces, and they can now start to aerosolize. They can start to move up with air currents. So if we go in within the next day or two and finally scoop the litter box, as we're scooping it, oocysts are aerosolizing and we can inhale them. And now we can get infected. Now, we are not the host that this organism wants, but it still can cause some issues in us. It won't make us chase after our cats. Don't worry, it won't do that. But what we've noticed is that it can affect things like women who are pregnant. It can cross the placental barrier. Just like what other organism can do that? Listeria, excellent. It can cross the placental barrier. Once it crosses, oh yeah, right there. Once it crosses, it can start infecting the baby and it can cause a miscarriage. This is why when women are pregnant, they get a pass on the litter box for 9 months. Isn't that nice? It's one of the big positives of pregnancy. Now, I will tell you this, if you have an indoor cat, you don't have to worry about it. You don't have to tell your significant other that either. You can pretend that you still can't do the litter box, okay? I mean, like, it works really well. The reason I wouldn't have to worry about it being an indoor cat is because my indoor cat's not out there eating mice that could be contaminated, right? And I probably don't have mice in my house. Now, here's where it gets interesting though. So we used to think that toxoplasmosis was only a risk to people that were pregnant. But we're finding something interesting now. This is what that paper in 2020 said. We're finding that some people naturally are more of a risk taker. Do you guys have a friend who's a risk taker? You guys are all nodding, yeah. So we're finding some people naturally take more risks. Maybe they like to jump off of cliffs in Hawaii. Maybe they like to skydive. Maybe they like to do, extreme sporting events. When we started looking at their antibodies in their body, we started finding that people who like extreme things tend to be positive for antibodies against toxoplasmosa. What that means is they were exposed to it at some point in their life. So we are starting to think that maybe some of the reasons that people like to do these risks is because toxoplasmosa has affected them a little bit, just like it does with that mouse, where it takes away their reasoning or risk assessment behavior. Isn't that fascinating? Something that we're just finding, and we're just scraping the tip of the iceberg, but we're just finding that it's something that seems to be present in our risk-taking group rather than our non-risk takers. People who are more careful tend to not show antibodies for this. So it's just kind of a fascinating study. You had a question, Ms. Brianna.
I was wondering if it takes over the cat's brain somewhere, how it does with the
It does not affect the cat's brain. That's a great question. So because of its life cycle, once it gets into the cat tends to be one of the final hosts. And then what it'll do is it'll pass through the feces. It needs to go through the intestine of the cat and then pass through the feces. And then that is now able to put it back into the environment where it can infect something else. It can pass on to a mouse or to another cat or whatever. But we don't see it affecting their brain. But again, we also didn't think it affected ours, and now we're finding that it might make us more risk-taking. So maybe if you have a cat that likes to live life on the edge, maybe. I don't know. If you have a cat who's like watching the cars and then runs across like right between, I don't know, like a little risk-taking, or jumps off your roof. I don't know. Like a little parachute, kitty parachute. It'd be kind of fun, right? Here's a picture of Cryptosporidium. So remember, this was our one that was responsible for that huge outbreak in which city? Milwaukee. Excellent. Our huge outbreak in Milwaukee. And so this is a picture of Cryptosporidium showing how it kind of just nestles itself into that intestinal lining, which again can cause inflammation and diarrhea. All right, our next group we're going to talk about are the Coeliophores. And Coeliophores are going to be ones that are going to have cilia around them for movement. We've talked about cilia in our lungs. So these are similar, those little hair-like extensions that help move them around. There's really only one in this category that's a human parasite, and that's one called Balantidium coli. So you thought Escherichia coli was a mouthful, and now you have Balantidium. So this is one that's going to get inside of our bodies, again, through ingesting contaminated water or food. Again, it's one that we don't have to worry about here. It's rare here because our water is filtered and it's taken care of for us. If it does get inside of our bodies, what it can do is it can cause colon inflammation. And that inflammation can get so bad it can actually cause colon perforation. So what does that mean? Yeah, it'll actually tear or open the colon. And part of that is because it produces a lot of gas as it grows. So it causes these massive CO2 bubbles in the intestine. And that pressure starts to push out. And as you can imagine, if you have any kind of rupture in your intestine, it can instantly cause a septic environment. Because all of that bacteria, all of that bile and stuff that's in there is going to get released into the body. So it can be potentially life-threatening. Again, something that we don't have to worry about here because in the United States, we are very fortunate with our clean water and our food. Let's talk a little bit about our next group. This is going to be the amoebas, so amoebozoas. And remember, our amoebas are going to move by those little pseudopods. So those little pseudopods that are going to be their little fake feet that we talked about before. Now, the amoeba that we used to worry about was one called entamoeba, and that caused dysentery, which again is intestinal upset. So think of like diarrhea. It can cause, again, swelling, inflammation. It can result in bloody diarrhea. So if we see bloody stool, we would want to check to see what it was. But now we have a new amoeba who's on the scene, who's fairly new, called Naegleria fowleri. You guys ever heard of Naegleria fowleri in the news? So this is also known as the brain-eating amoeba. Have you heard of that in the news? Yeah, we see more nods. Okay. So Naegleria fowleri is one that we find primarily in humid states. So in Texas, in Atlanta, in Florida. We haven't had a case here in California yet. We're not humid enough. Or at least that's what it seems to be. So they're found in human environments, and they tend to be associated with lakes, a lot of times ditch water, like some kind of runoff water, and unfortunately, a couple of times in water parks. They were able to slip into water parks. The reason we worry about them is if you get water sprayed into the nose with force. So picture like if you're water skiing, you get water kind of pushed into the nose, wakeboarding, or if you are at a water park and you're going down a water slide. If you have water being pushed in the nose with any kind of force, and if this amoeba is present, it can move from your nose through to the inner brain lining. And once it gets into the brain, it can start consuming or eating the brain. You'll actually see tracks through the brain where the amoeba is moving. Now, the reason we're so concerned is because of the death rate on this. What's the death rate? 97%. Yeah, we've only had one person that recovered completely and one person who recovered who's still having to seek a lot of medical treatment. So that's like 2. That's it. And both of them were young. They were under 14. And we think that one of the reasons they survived is because their brain tissue is so young, it was able to try to develop around this. So what does it cause? It causes massive inflammation in the brain. Because this organism is consuming the brain, it actually causes loss of brain tissue. And it unfortunately results in the body just shutting down. And again, because it's an amoeba, it's similar to us, so we can't just put a whole bunch of antibiotics in there. And we can't really treat it too well. We can just kind of do preventative. We can try to help the person and see if their immune system will help them recover. But there's not a lot of treatments as far as treatment options that we can do. Again, most of the cases have been in Florida and Texas. And what's really weird about this is if you're exposed to water that has this amoeba, but you're not using any kind of force with it. So in other words, if I just go to a lake and I swim in the water just like normal, I can swim in water full of this amoeba and not get it. It has to have pressure to get up inside that nasal and to the brain passage. So it has to have a pressure associated with it. So just a really fascinating, odd organism. So Mishala said, what about netipots? That is a great question, Mishala. So on Zoom, we have, what about netipots? There was one incident of a person who used well water in his netipot. And his well, unfortunately, had these amoeba in it. And so if you read your netipot's information, it always tells you to use what kind of water? Distilled, yeah. So water that has been distilled, purified, ultra clean, not well water. Okay, well water hasn't been treated. That's like basically taking Lopez Lake water and pouring it in your nose. We don't want to do that. So what's going to happen then is he ended up putting it into his netipot. He was treating his nose, and if you've never seen a netipot, it basically moves through the nasal canal. You put saline water in the nasal canal, and it kind of flushes things out. And unfortunately, the well had a high level of these amoeba present, and it ended up contaminating himself. And he was also using a syringe to also help in addition to the netipot, and that syringe had enough force to push the amoeba into that inner brain barrier. So the question is, will it work with netipots? Unfortunately, yes. So make sure you use distilled water in your netipots. Okay, one of our last ones we're going to talk about are the euglenas. And the euglenozoas are ones that look very delicate. They almost have like a worm-like appearance to them. They're really beautiful to watch when they swim. They kind of have like this little undulating swimming appearance. But again, they could cause a couple of important diseases in humans. They are transmitted similar to malaria through insect bites, but malaria was transmitted by mosquitoes. These tend to be transmitted by biting flies or by biting bugs. Luckily, we don't have the flies here, so we don't have to worry about the type of fly that spreads them, but we do have the type of bugs that spread them. So let's look and see how these are. The first illness that can be caused by one of our hemoflagellates that we're looking at is going to be one referred to as African sleeping sickness. The organism that causes it is trypanosoma. Think about, you're going to go tripping on sleep. It's trypanosoma, an easy way to remember it, trypanosoma. What happens is if you get bitten by this little fly called a tsetse fly, I'll go to this next picture. This is our little organism, trypanosoma. If you get bitten by a tsetse fly, it can carry trypanosoma in its saliva. And when it bites you, it'll pass this organism to your body. What'll happen then is this organism will start to reproduce inside the body. And when it reproduces, it produces toxins that start to make us feel tired and worn out. It also starts to steal our ATP. And remember, if we don't have our normal amount of ATP, we feel tired and worn out. As this happens, then we start to enter what's referred to as sleeping sickness. We start sleeping up to 20 hours a day. 20 hours a day. That's a lot. I mean, I know that sounds kind of like the dream right now because we're all so busy. It's like we can't even imagine 5 hours of sleep, much less 20. But imagine that happening day after day after day. You know, you're just completely missing parts of your life because you're sleeping through it. So with the trypanosoma, there is treatment for it. The problem we run into is the areas that's found in, which is usually in Africa, they have so many of the tsetse flies that even after we treat somebody, they can get infected right away again because these flies will immediately come and bite them again. So it's like this constant cycle. So we used to try to attack the trypanosoma. We're changing our approach now. We're trying to attack the flies. So we're releasing sterile flies. That's one thing we're doing. We're doing that with malaria as well, releasing sterile mosquitoes. The idea here is that way these organisms are out there, but they're sterile, so they can't reproduce. So we're hoping that we can start knocking their generations down, getting their numbers down, and that's what we're working on now. Now, I mentioned they can also have, some of the organisms can also be spread by bugs. So there's another type of trypanosoma that's referred to as Chagas disease. And Chagas disease is also called the American trypanosomosis. And so what happens here is instead of being spread by biting flies, which we don't have in the America region, south or north, they are spread through a beetle. And so here's a picture of the beetle. It's called the triatomine bug. It's an orange and black beetle that is referred to as the kissing bug. And the reason it's called that is it's a nocturnal bug that comes out at night when we're sleeping, and it's attracted to CO2. So when we're breathing out in our sleep, it follows our CO2 trail. And then what it'll do is it uses the CO2 to hone in on its victim, and then it will bite and take a blood meal from that victim. Now, since it's used CO2 to hone in on us, that means that we usually end up with bites around the mouth or around the nose, which is why it's called the kissing bug, is because usually it's around our face where the bites are going to occur. So you'll wake up in the morning and you'll have these bites. Sometimes they can bite near the eyes or the nose. It's somewhere on the face. And they can cause inflammation and irritation. Now, that seems bad enough. But if the bug is infected with one of these trypanosomas, it can also put that trypanosome in your bloodstream. And so then that can start making you feel tired, not as bad as sleeping 20 hours a day, but you feel tired and you feel run down. So do you think that we have Chagas disease here? The answer is yes. So Chagas disease started primarily in South America, and it's been kind of working its way up through Central America, and then it hit the Mexico region, and now we're marching its way across the U.S. So do we need to be worried? Well, think about all of us here. Do we know of anybody who's ever had a kissing disease, Chagas syndrome? Yeah, everybody's like, no. Yeah, so again, why not? Well, it tends to be more common in South, Central America, and in Mexico because of the types of homes that they have. They tend to have adobe-style homes. They tend to be loose bricked, which means there's lots of opening for bugs to get in and out. Do we have homes that are pretty open to bugs? Not really, right? And what do we do if we see a bug? We kill it and we call an exterminator, right? We go into panic mode. Yeah. So we tend to be much more able to, treat our homes with insecticides or pesticides. Our homes are not as open because we use, different types of building material. And so because of that, we tend to keep the kissing bugs out. So even though they're here, even though we're seeing cases being reported, it's not like it's a huge number. And it's not like the alarm has been sounded yet. And that's why, is because we're just not that concerned yet. The reason we are monitoring it, though, is to make sure that the numbers don't go up. If the numbers go up, then we'll have to look at a way to eradicate these little beetles and get rid of them. Okay, our last little group we're going to talk about are the helminthes. And helminthes are going to be parasitic worms that live in our environment. Now, these worms lack a true digestive system. They have pretty reduced nervous systems. but they have very complex reproductive systems, which means a lot of times they need to be in multiple hosts or move back and forth between hosts to be able to reproduce. We have two groups in here. We have the platyhelminthes and the nematodes, and we'll talk about, again, the ones that affect us. Again, lots of different organisms out there, but we're just talking about the ones that can cause illness. So the platyhelminthes, these are going to be the organisms that you might have heard of referred to as flukes or tapeworms. You guys ever heard of those before? Yeah. So these are ones that we can ingest eggs of the tapeworms, or we can get exposed to flukes in contaminated water. They move inside of our bodies. They tend to move to our intestinal regions, and then they start to grow in there. The tapeworms we have to be really careful of, because whenever we have defecation occur, the tapeworms can come out with our fecal deposit as well as eggs, and then that can be an easy spread from one person to the next. If they're not careful, if they're changing diapers or something, not washing their hands, they could potentially get infected that way. There was actually a dietary aid that was sold in the 50s that was helping people lose a lot of weight. And then people started reporting that they were feeling tired. Stefani was like, nodded. She remembers the story. Yeah, they started feeling tired and they weren't feeling good. They went to see their doctors and they found that they were full of tapeworms. And the doctor was like, how on earth did you get all these tapeworms? So they started trying to review what they'd been eating. They found out that the dietary pill they were taking was full of dried tapeworm eggs. So that was what the producer of the company was using to help you lose weight. It was like, here's your parasite. You eat food, you get half, and your parasite gets half. So as you can imagine, you lost weight quickly, but you also lost nutrients and energy and everything else that goes with that. Another one that can affect us are ones referred to as roundworms. And so these would be ones that we hear a lot of times with our animals, right? So our animals can carry tapeworms, our animals can carry roundworms. We worry, like with puppies and kittens, we have to treat them for roundworms, right? And you might have wondered, why do puppies and kittens always have roundworms? A lot of times they get them from their mothers. So when they're still developing in the womb, they can get exposed that way to the eggs. Also, they're carried by fleas. The fleas, when they are biting them, if they're infected with a roundworm, they can bite the litter, and then they can infect them that way as well, which is another reason to keep our fleas under control in our house. So usually there are common ways of getting exposed to these organisms is through ingestion of eggs, whether that be in feces or contaminated water or eating undercooked meat. We had an outbreak of a type of worm recently in Hawaii, and you probably heard it in the news, the rat lungworm disease. Do you guys remember hearing that in the news? It was we've had so much news. It was in the end of 2019, but then we had that thing COVID come out, and everything else just kind of disappeared, right? So we kind of forgot about that. But yeah, so rat lungworm, it's still present in Hawaii. And what it is it's a little worm that's carried in snails. And these snails are really tiny, like the size of a pinhead, really, really tiny. And these snails ended up getting imported accidentally from Thailand into Hawaii. And so when they were imported on lettuce leaves. And so if you ordered a salad when you were in Hawaii, if it wasn't properly washed, or if you bought produce at the store and it wasn't properly washed, it could have these little tiny snails on it. Now you might think, oh, I would definitely see it. But picture the size of a pinhead snail, really tiny. It would be really hard to see. And if you dice up your lettuce and throw them into your salad, it was easy to be consumed. And so unfortunately, these little snails had snuck in. Nobody knew they were there. They started having tourists and people eating, lettuce, and they had the snails on them, and they ended up ingesting these worms. And these worms, rather than moving to the intestines, they also can take residence in the lungs. And so it can cause a respiratory infection. Again, it's something we can treat, but it's something that was just unexpected in Hawaii. We just didn't see that coming. So now they are much more careful with their screening of their lettuce. But still, if you order a salad there, look for snails. All right? I have family that lives there, and I'm always teasing them. I'm like, check your lettuce, check your produce. Be really careful. Look for these little guys. Because now that they're present, they could potentially, keep causing disease. Any questions on what we just covered with our eukaryotes? Okay, so it's a lot of information. So how should you study for it? Should you wait till the night of the exam? No. Should you start reviewing now? Okay, and what do I usually recommend when you're studying? tables, right? I recommend tables. So start putting together your tables now. Start putting together your information now so that way you can start to build the tables that you need to know for your quiz and for your exams, okay? Again, use your homework as a study tool. So don't do your homework quite yet, start studying, and then do your homework as kind of a way to quiz yourself to make sure that you're understanding the material. Okay, so I'm going to share my next screen with you, and we're going to switch gears here. We're going to start talking about our viruses, which I know everybody's excited to hear about. All right, I got my chat screen up. So as usual, let me know in Zoom if you have any questions. And the first thing that comes up with our viruses is trying to figure out how to characterize viruses. And one of the big questions we face is, are they really dead or alive? And that's kind of an odd question. But it's because of the fact that viruses don't really fit our criteria of living cells. One of our criteria to be living is that you have to be able to reproduce on your own. And viruses can't do that. They have to have a host to reproduce. So we have like this big debate always going back and forth about whether they're really living cells or not. What we do know is that viruses have been around for a very long time and that they can cause some really bad illnesses in human populations. So the name virus literally means poison. So it's something that is poisonous or toxic to us. And that kind of makes sense when we think about having a flu virus. We think about, not feeling well. And it does feel like you have something toxic inside of you. A lot of biologists, as I said, still consider them to be non-living. You'll have a few virologists that are like, no, they're living in a different way. But the reason they consider them to be non-living still is because of the fact that they are not able to reproduce on their own. In fact, they also don't have any organelles. They don't have ribosomes. They can't make their own ATP in any way. And they have to have a host to be able to survive. So that's a lot of limitations. And yet, they have been successful since the beginning of mankind, which is pretty fascinating that we have so many viruses out there. They're so simple. They're composed of two parts. They have a nucleic acid, and again, that nucleic acid is going to be either DNA or RNA. So it's going to be one or the other. They never have both. So think about our cells. We have both, right? We have DNA and RNA. In this case, they always have one or the other. Now, to protect their DNA, they have to have some sort of coating around it, or to protect their RNA, they have to have some sort of coating. And so, in addition to a single nucleic acid, they are also going to have a protein coat. And that's referred to as a capsid. It's basically going to encircle or encapsulate their DNA or RNA. So this little capsid or protein coat protects the nucleic acid. Sometimes you also can have viruses that go an extra step, and this is just in a few of our viruses, that produce a second protein coat called an envelope. So I have a nucleic acid, I have my protein coat, which is a capsid, and then some produce another coat on top of that, which is referred to as an envelope. And those, as you can imagine, sometimes can be a little trickier to treat because I have to break through two layers to try to get them to go away. Now, viruses are spread through a lot of different ways. We can get exposed to them through the wind, through the water, through food, through blood, direct contact, talking to people. One of the hardest things when we have a new virus appear, just like with COVID, is trying to figure out how they're spread. We originally suspected that COVID was spread through the air because it was spreading so quickly, but we also thought that it was probably living on surfaces. And that's why we had that massive organization to, try to clean every surface, wipe down every surface. But further research, because we're still learning about COVID, has shown that it really doesn't live very long on a surface. Meaning if somebody coughs on a table, there's not a lot of nutrients on that table to support it. And as we know, they can't make their own ATP, so the COVID tends to dry out and die quickly. So it doesn't tend to have surface spread, which is good. That's really good for us. The other thing about viruses is they are very, very tiny. And so they actually make bacteria look like monsters. Think about like our E. coli. Remember how tiny it was looking through our microscope? We could barely see it. E. coli will look like a monster to a virus. That's E. coli at the bottom. See how big it is? Remember, we were straining to see it. And then look at this little adenovirus. I mean, it's up towards the top, the little star-shaped one. Look how tiny it is, 90 nanometers. I mean, that thing is microscopic. So these are things that we're going to need to see with like electron microscopes. These are things that are going to be so small. We're going to need a lot of help to see them. In the corner, looking like a big red blob, that's our red blood cells. So, I mean, our red blood cells are not that big, but look at, they're like massive compared to our little tiny viruses. And then just looking at this picture in general, do you see how viruses can have different shapes? So they can have a lot of different shapes or structures associated with them, depending on which virus we're talking about. Now, the interesting thing about viruses is that they are very specific. And so they're going to have a specific host cell type that they can infect. For example, if you think of a cold, what symptoms do you think of with a cold? Runny nose, watery eyes, congestion, right? So a cold virus is able to infect cells in the respiratory region, the nasal region. What we don't think of is upset stomach or diarrhea with a cold because it's not able to infect our intestinal cells. It's not able to infect those cells in our bodies. So the way this works then is every virus that's out there is going to have a specific type of cell that it can attach to or that it can try to match up with. And the idea then is that when it finds that cell and attaches to it, it can put its nucleic acid, whether it's RNA or DNA, inside of that new host cell. But again, they can only attach to certain cells. And this is why we picture certain areas or certain regions located with certain viruses. So influenza viruses will infect respiratory tract. Polio, when we talked about before, affects nerve cells. Even plants have viruses. There's a plant out there that is the tobacco plant, and one of the first viruses we studied was the tobacco mosaic virus, which is a virus specific for a tobacco plant. So every organism that's out there can have their own viruses that can infect them. Here's a picture kind of explaining why viruses can only attach to or infect certain cells. So a lot of our viruses, as we saw in that previous picture, where we saw that red blood cell and the E. coli, and we saw their different shapes and varieties they can have. Well, those different shapes that they have can affect what kind of surface proteins they have. And these surface proteins can affect who they can attach to. It's kind of like how our enzymes had a lock and key. The same idea. It's like a lock and key. So the viruses can only attach to the cells that fit them. And again, this is why we only see them in certain regions of our body. This is also why we don't worry a lot of times about getting viruses from our dogs or our cats, because they also tend to be host specific, meaning that they will only fit with a cat cells or they'll only fit with a dog cells. They don't fit our biology. This is also why COVID was so surprising to us. What do we think that the original infectious organism was with COVID? A bat, yeah. So we think it started with bats and that it was able to mutate and move to humans. That's something that's pretty rare for a virus to be able to do that. We see that occasionally. We had that happen, for example, with swine flu, H1N1. It started with swine or pigs, and it was able to move to humans, and now it's actually established itself as a typical flu strain. So we do see it happen occasionally. It's a mutation that can occur, but it tends to be rare. So who has viruses? Well, as I mentioned, plants can have viruses, invertebrates can have viruses. So, I mean, even worms can get viruses. Protozoans and fungi that we just talked about, they can get viruses. And guess what? Even bacteria, even the things that make us sick can get viruses. That is called karma. So bacteria can get attacked by viruses that we call bacteriophages. And the bacteriophage, which is sometimes just shortened to a phage, that's another name that we use, is something that can go after our bacteria and destroy them. We are really excited about bacteriophages. We're going to watch a video in here in a few weeks about resistant microbes. So bacteria that are resistant to every antibiotic that we can throw at them under the sun. We are starting to come towards the end of the golden age of antibiotics. And what that means is when penicillin was first developed, we were able to wipe out organisms for the first time. And we've had all these great antibiotics that we can use since then and that we've developed since then to kill our microbes. The problem we're running into is the bacteria have figured it out and they're mutating and fighting back. So one of our big defense lines that we're looking into right now is the use of a bacteriophage to treat bacterial infections. So in other words, introducing a natural enemy, a virus in our body that won't attack our cells, but will attack the bacteria. And it's actually working. We have certain strains out there that are resistant to any natural or chemical antibiotic. When we put in a bacteriophage, it will destroy the organism. So this is very exciting for us. So a lot of research is being done here. Now, as far as how we can categorize viruses, because you know we're biologists, we like to sort, we like to organize, right? We admit we have the proteista jumper, but other than that, we like to sort and organize. And so the way we can sort and organize our viruses is based on what kind of nucleic acid they have, whether they have RNA or whether they have DNA. That's our first categorizing that we can do. So DNA viruses, the beauty of a DNA virus is it tends to be very stable. And what this means then is if it's a stable virus, I can easily make a vaccine for it. So if I have something that's not mutating all the time, I can make a vaccination process. I can give people, immunity with a vaccine or at least some sort of protection. And so a lot of our DNA strains that are out there are ones that we can treat preventatively through vaccination. So we mentioned smallpox. I told you smallpox is the only type of microbe infection that we have eradicated or eliminated off of our earth. So smallpox was luckily a DNA virus. And because of that, it was very stable. And so we were able to do a mass vaccination program and get rid of it. Hepatitis B, that is also a DNA virus. So if you travel out of the country, remember, I always tell you, go to CDC, see what vaccines they recommend if you're going to travel somewhere. And one of the ones they recommend is if you leave the U.S., get a hepatitis B vaccine. Plan ahead of time. Hepatitis vaccines, they take about 6 months. You have to get a shot, wait 6 months, and then go back. So they're kind of like COVID, where it's a sequence, like our sequence vaccines. So plan ahead of time. You don't want to do it like the night before you leave, okay? Because you need to make sure you get that second shot. RNA viruses, these are ones that are difficult. They tend to mutate very easily. They tend to be much more difficult to try to get ahead of with a vaccination program. And then when we do have a vaccine in place, they mutate again. So one of the ones that we've been encountering is COVID. COVID is an RNA virus. That's one of the reasons why we keep saying that we're going to probably have to have booster shots or vaccinations moving forward, similar to what we have with the flu vaccine, where we're going to have to get administered vaccinations constantly because of mutations. That's also why the flu always has to have shots every year, is because of the fact that it can mutate or change. We have different strains that can come around. One thing we run into with the flu vaccine is it takes a long process to prepare the vaccination. And sometimes when we prepare it, we might not have the exact strain that's going to be circulating that year. And so the flu vaccine can have different effectivity each year depending on which strain is going around. We try to guesstimate based on what data is present as to what flu strains are in the area. But again, it's kind of a guess. We have down here that they're single-stranded, as I mentioned, but we also have that they're plus sense or minus sense. And you might be going, well, what does that mean? Well, it turns out that some RNA comes ready to go. We call that plus sense. And what that means is as soon as it gets into a host cell, the RNA goes directly to the ribosome. And then it can start being read. So remember, that would be translation. It can start being translated in the ribosome right away. So that would be a positive sense RNA. Some RNA comes and it's not ready to be read in the ribosome. It actually is going to have a fresh strand of mRNA based on it. So what it's going to do is it's going to use our RNA to make a copy of its strain. And so then it'll use our RNA to go to the ribosome. So it uses itself as a blueprint. It's just that it can't go directly to the ribosome. It has to have us first. Usually that's because of the fact that it needs to have something happen because the mRNA strands that it has in place right now would be unstable in our ribosome. So it uses rRNA to make its own ribosomes. Okay, so we have nucleic acids in, whether it's RNA or DNA, that will classify them. The next step that we can use to classify them is their morphology. Do they have an envelope? What is their shape? What do they look like? Size? That can also help us classify them. And then the final way we can classify them is through how they replicate. So what kind of hosts they need to be in. We put that at the end because we know that they can occasionally switch hosts. So we make that kind of a third criteria, but we want to know what hosts they can potentially impact. So as I mentioned, viruses really are not considered to be living because they're so reproductively limited. So again, if they can't be inside of a host cell, they're not going to be able to survive very long. We talked about viruses living on surfaces, and I mentioned that COVID doesn't live very long on the surface. Other viruses that don't live long on the surface would be things like the flu or things like a cold virus. They don't live very long on a surface. Herpes virus, they don't live very long on the surface. They have to be in a host as soon as possible. Because of the fact that they need a host, they're considered to be obligate parasites. They live on the host. And because they're found inside of us, they're intracellular. So obligate intracellular parasites. So what do they look like? We mentioned that we can classify them based on structure, and they have three main structures associated with them. There's the polyhedrals that kind of look like almost like a little stop sign. There's the helicals that look like little coils. And then there's the complex, which are my favorite, that look like little spaceships. They look like something like out of an alien movie. So I think you guys are going to like this. So let's take a look. Okay, so here's our polyhedrals. So polyhedrals, again, they kind of look like a three-dimensional stop sign. So if you picture the little angles and then kind of like a 3D look to them, they're going to have inside of them either RNA or DNA. And all those bubbles that we see, those blue bubbles in this picture, that's all their protein capsid. That's their coat that's surrounding their nucleic acid. And again, its job is just to protect the nucleic acid inside. Some of the viruses can have that additional protein coat, which is the envelope, and some won't. It just depends on the virus. If they do have an additional protein coat, a lot of times they will be spiked. So let's think for a minute about our COVID virus. Does our COVID virus have spikes on it? Yeah, that's actually why it earned its name corona. The co part of COVID is short for corona. So the reason it is the coronavirus or COVID virus is because it has a crown of spikes around it. So you've been seeing pictures of it in the news. So you've been seeing a polyhedral virus with spikes around it. So it has that extra layer. Here's a picture of a polyhedral virus. So does this kind of look similar to what we've been seeing in the news when they flash up our COVID? It usually has more spikes. COVID has even more impressive spikes. This is an example of a mastodinovirus, which we'll talk about in a few slides, but it's one that can cause gastrointestinal upset, so kind of like a flu-like syndrome, as well as a respiratory tract. It depends on where we first get our highest number, meaning that if we ingest the virus, it'll move to the intestinal tract. If we inhale it, it'll stay in the lungs. So it just depends on our process of exposure as to whether or not it gets in different regions of our body. The next one is the viral group of the viruses. The helical group of the viruses. And the helical group is going to be ones that are coiled or have some sort of a bend to them. So a lot of times we'll see pictures of Ebola, for example. And the reason Ebola is a virus that's used a lot for pictures is because it's so big. It's easy to do a quick snapshot of it. And what happens with our helical viruses is they have a coil or a bend to them. And as you can see, Ebola has this kind of little bow tie effect. The first time virologists saw Ebola, they said it wears its bow tie to dinner. Isn't that just kind of like a dark humor? It's wearing this little bow tie. But yeah, so it's always got kind of that little front bow tie, which makes it really obvious. These organisms, when they're helical, they can be DNA or RNA, so they can be one or the other. They will have a capsid present, a protein coat, and they may or may not have an envelope. So they could potentially may or may not have an envelope. Ebola is one that is going to have an envelope present. We can see this dark outline around the virus. Do you guys see that dark black line? That's its little envelope that is coding it. It is also an RNA virus, which is one of the reasons why it has been hard to develop a vaccine for it. It's hard to, make something for something that's constantly going to be changing on us, and RNAs change a lot. With that envelope, it is also harder to treat and to get rid of, and that's one of the reasons why Ebola can be so dangerous for us. What class was Ebola in? Do you guys remember? What was our biosafety level? 4. Yeah, it's a BSL-4, which remember is the top, right? The BSL-4s are the top, which means no vaccination, very little treatment options if you get exposed. In Africa, they had an outbreak from 2013 that lasted until 2016. And it unfortunately is still going on at a low level. So Ebola never really goes away 100%. It still occurs. I've had students ask me in the past, why does Africa always have the viruses? Why do they always have the tsetse flies? Why do they always have the scary parasite? And the reason is, where did humans originate? What is our oldest ancestry that we can follow? To Africa, yeah. So that's where we've been the longest, which means organisms living there have had more time to adapt to our biology. So that's where, mankind originated. And so viruses that are dependent on them also originated there. And organisms like, different types of parasites originated there. We were just a new host. So it's not like Africa's bad. It's just that it's, an area of our origins for all of us. Okay, here's my favorite one. This is the complex virus. Does this look like the cutest little spaceship ever or what? Or maybe like a little alien? I don't know. It's so cute. So these are ones that we see a lot of times in the shape associated with bacteriophages. So again, these are ones that would attack bacteria. Maybe that's another reason I like them too, is because they're helping us. So they have these little cute little capsid kind of heads that look almost polyhedral. Then they have these little bodies that are in the middle that look kind of helical. So it's like this mixture. I've got this little polyhedral and this helical region. And then I have these little legs that are going to attach, like a little tripod that'll attach to my host. What I'll do is when I crawl around on a cell and attach to a host, I'll find an area that I like that I can fit on, my little tripod can fit on it. I will contract that helical region kind of like an injection, and I will insert the nucleic acids that are kept inside of my polyhedral-like head. So it's like this little compression that it does. And so it inserts the viral component inside, which is pretty cool. Again, they may or may not have envelopes present, but they always have a capsid present to protect them. Okay, we're going to go ahead and stop here because the next time we're going to pick up on some examples of viruses. So this is a good stopping point for us, and it's also a chance for us to review for our labs. Okay, so in our nitrate broth, we have a Durham tube. What does that mean we should look for? Gas. Gas. So hold up your broth and look to see if you have any gas bubbles. Gas bubbles are going to indicate nitrogen gas, or N2. Do we see any bubbles at all? Okay. So if I don't see any gas, then that means that my organism was not able to take nitrate and go to nitrogen gas. Is there anything else that could convert the nitrate to based on your reading? Yeah. I could also have nitrate convert into NO2, nitrite, right? So we had that in our intro. Okay, so if I have nitrite present, how would I be able to tell? We're going to add some reagents to see if we can turn red. So on each side of the room, we have some reagents. We have nitrate reagent A and nitrate reagent B. Do you think it matters what order they're added in?
Yes.
Yes, it always matters, right? Okay, so we're going to do A first. A is number 1. We are going to add 5 drops of reagent A. We're gonna wait about 2 seconds, and then we're gonna add 5 drops of reagent B. Go ahead and go do that. Yeah. Okay, we should be seeing 2 tubes that are turning red. Who is our organism that is turning red? E. coli. E. coli. Does everybody see that E. coli is our positive here? So what that is indicating is that E. coli is going to be positive for the nitrogenase enzyme we're testing for. Now we've said before that E. coli can grow with or without oxygen. So what is it? Facultative anaerobe. Excellent. It's a facultative anaerobe. This should make sense then. When oxygen is present, it can use that as its final electron acceptor. If I put it in an environment where oxygen isn't around, it can still grow really well because it can switch to nitrogen. And nitrogen can be its final electron acceptor. So what's happening here then is I'm going from nitrate to nitrite. The nitrite is reacting with my 2 reagents and it is creating a pH color change that I can see that is causing that red reaction. Okay, so right now E. coli looks great, but we still don't know about Acinetobacter. We have another question we have to answer. And that question is, okay, we know Acinetobacter does not have nitrogen gas. We know it does not have NO2. So the question now is, does it still have nitrate sitting there unused? Or did it convert it to something we haven't tested for, like ammonia or something along those lines? So the last test we're going to do is to only your Acinetobacter, to one of your Acinetobacter tubes, just one. You're going to add a little bit of zinc, okay? So we have some zinc dust over here. You're going to use your spatulas that are on the side or some on that side of the room too, and add a few sprinkles, just a few sprinkles of zinc dust. So if we see a red color occur with zinc, then that means that the zinc is binding with the nitrate, and that's going to produce a red color. That means our organism is completely negative or positive? Negative. Negative. Excellent. So we're going to check and see. So go ahead and add some zinc to one of your Acinetobacters. Is nitrate still present? No. No, the nitrate's gone. So that means that by that definition, my A. baumannii is actually positive. And how do we know this? Well, it didn't take nitrate to nitrite. It didn't take nitrate to nitrogen gas, but it took the nitrate to something else because there's not any nitrate to play with the zinc. If the nitrate was still there, it would interact with the zinc, we would get a chemical reaction and they turn red together. But the zinc is not doing anything. That means it doesn't have any nitrate to play with. So since I don't see a red coloration, that means that something else happened. So that means I could have what? What might have formed? Ammonia. Ammonia. Yeah, I probably had ammonia form in there. So I had some sort of an ammonia composite. As it sits for a while, the zinc will start to interact. That's why you have to read it right when you're at it. So don't worry, it'll sit for a while. Yes. Part of that is from oxidation, just from being exposed to the air and being shifted. So you will see these colors change with time. All right. This is why we have to read them out right away. Right? Okay. So does everybody understand what's going on with the nitrate test? Okay, we understand that they were both positive. Aaron, what's that?
Oh, you said they're both positive.
They are both positives for different things. E. coli was positive for nitrite, and Acinetobacter baumannii was positive for ammonia, but they are still positive because they're producing the enzyme nitrogenase. They have to produce that to break that nitrate down.
Nitrate reductase.
Yes, you got it. Okay, so that gets us through our nitrate broth. How about oxidase? Let's do that one next. So at the end of each of your benches, you have a little white tube. And if you don't want it to wait for it to make its rounds, you can also go to the side. I think we have some on the side. That are oxidase reagent strips. All right, so what is oxidase looking for? Remember we said we were looking for a little group in the ETC. The electron transport chain. What was the name of that little group?
Cytochrome C.
Excellent. Cytochrome C. So remember, we're looking for a little group of electron acceptors and passers. These are ones that are going to get electrons and then pass them on towards oxygen or towards nitrate, whatever is going to be our final electron acceptor. But to do that, this little group is one that will have specific proteins in place, and we're going to see if these specific proteins are in place to react with our oxidase reagent. Now, I told you last time it's kind of like having a little bit of columns like A, B, C. If I'm an organism that is oxidase negative, I might have maybe Z, Y, or X, Y, Z. I don't have A, B, C. I'm looking for A, B, C specifically for the exact set of proteins. So, how am I going to do this test? This is probably the easiest test you're ever going to do in here. You're going to get one of your little oxidase strips out. Again, they're at the end of each of your benches. You can pass them down from one another, or you can go to the side of the room if you don't want to wait. On your oxidase strip, you're going to see that they have this little white coloration and a clear tab at the top for you to hold on to. And what you're going to do is you're going to take this strip and you're going to drag it right across the surface of your Petri dish with your organism on it. So we have two organisms on our plate. We have E. coli and what's the other one? Pseudomonas aeruginosa. So I'm going to need two strips, one to drag across E. coli, one to drag across Pseudomonas. How fast do I need to read these out?
10 seconds.
15 seconds. So remember, in your lab intro it says 15 seconds. I need to read between 10 to 15 seconds. So what I'm going to look for is I'm going to drag these across the colony. If I see a blue coloration within 10 to 15 seconds, that means I am oxidase positive, which means I have the ABC portion of my cytochrome C. I have my proteins in order. So go ahead and try it. Get your strips. You need 2 each. Get your strips, and you're just going to touch them to the colonies and drag them down the plate to get like a little paste on here. Did everybody see the Pseudomonas produce a pretty blue coloration? How about E. coli? Did it produce any color?
No.
No. So no color change. Now, if you let that E. coli strip sit too long, it'll start reacting with the oxygen of the air and you will get a blue color change. That's why you have to read it right away. It has to be within that 10 to 15 second zone or else you get a false positive. So on here then, what we see is our Pseudomonas is positive, our E. coli is negative. Okay? So what that means again, Pseudomonas has cytochrome C, a network of proteins that are going to pass electrons along. E. coli does not have this exact same configuration or network of proteins. It has a different network. So these would be like ABC, these would be like XYZ. Okay, they still function, just a different network. Guess what? We have cytochrome C as well. Ooh, that's exciting. And what do we share with Pseudomonas? What does Pseudomonas like in regards to oxygen? Does it need it or does it not?
Pseudomonas.
We've had a couple labs with it. What happened when we put it in the FTM broth, the fluid thio? Where did it grow? Was it at the bottom or at the top?
The top, I think.
At the top. Okay. So if I'm growing at the very tippy top of a broth, what does that indicate about me?
Obligate aerobes.
Obligate aerobes. Is that what you were going to say too? Yep, got it. Obligate aerobes. So our Pseudomonas has the same network of enzymes we do, the same proteins in place. And just like us, it is an obligate aerobe. If I take oxygen away, it's not going to be happy. Okay, our last test that we have to do is the catalase test. So to do this one, you're going to take a slide, a blank slide. You're going to put a drop of catalase reagent, which is hydrogen peroxide. And then you're going to take a plastic loop. We have plastic loops at our desk. And the reason, do you guys have plastic loops? Yeah, they're right here. They're on your tray. The reason we use plastic loops is because the metal loops sometimes can interfere with this reaction. The metal can actually react. So what we're going to do is we're going to put a drop of our hydrogen peroxide on the slide. We're going to do two because we have two different organisms that we're testing. And then we're going to add one of our plastic loops full of our bacteria that we're testing. And what are we going to look for?
Bubbles.
Bubbles. We're going to look to see if we see bubbles. If we see bubbles, that indicates that we are catalase positive and we're able to break down toxic forms of oxygen. So with that in mind, let's go ahead and do our test. So you should have, so I see a couple people are doing their catalase test. Remember, you're going to put a drop on each side of your slide and then use your plastic loop to add your bacteria and see which one of them bubbles. Which one bubbled for you? Excellent, good job. Okay, let's talk about our setup. So put your plastic loops away with a false alarm. We'll come back to this. As far as our setup today, our new lab for the day is to look at our chromogenic media. We mentioned chromogenic media is going to have a sugar attached to a chromophore that has color on it. Depending on which sugar they prefer to eat, the bacteria will turn the color of the chromophore that's attached to it. So let's pretend that I only like to eat glucose, and glucose has a chromophore attached to it that is blue. As I eat more and more glucose, I will become stained, kind of like when you eat a popsicle. If you eat more and more of that popsicle, your tongue turns the color of the popsicle. And so the next time when we look at the bacteria on our plates, they will be stained different colors depending on what sugar they liked. So I'll have glucose in there, I'll have sucrose, maltose, fructose, all sorts of sugars. And we'll be able to tell, based on the color, who likes to eat what. So what do we need to set up today? Well, today you're going to be setting up a bacterial UTI plate. Remember, UTI stands for urinary tract infection. These are the common organisms that cause UTIs. Which one's the most common up here?
E. coli.
Excellent. E. coli is always number one as far as the UTI count goes. So we've got E. coli, Staph aureus, Citrobacter fungi, E. faecalis, and the new one for us, Staphylococcus saprophyticus. Staph saprophyticus is similar to our Staph epi and our Staph aureus. The difference is that Staph sapro is a little more like Staph epi, meaning it's opportunistic. So if it gets into our bladder, it will cause an infection. If it's on our skin, it doesn't hurt us at all. Okay, so that's the difference, just kind of like how Staph epi is. Okay, so you're going to separate your plate into 5 different areas, and you're going to inoculate with these 5 bacteria. We're not looking for isolation, so what kind of streak do you think we're going to use? You got it, the fishtail. So remember what that means. Fishtail, there we go. We're going to tap, and then what am I going to do? Line and a zigzag. Excellent. Line and then zigzag. Excellent. Do I cross over into the other bacteria's boundary? No. Be really careful, because if you do, your colors will bleed. Okay? We don't want to do that. We want to be able to see the individual colors that each one creates. All right, so we have 5 different organisms we're going to be putting on this plate. When we're done inoculating our plate, where is it going to go? In the incubator. Yeah, we're going to incubate it at 37 C for body temp. Okay, in addition to our bacterial plate, we are also going to set up a candida plate. Remember, we talked about a candida that is an emerging infectious disease. Which one was it?
Candida.
Did I hear the whisper? Auris. Excellent. Good job. Yeah, Candida auris. Candida auris is an emerging infectious disease. It's one that we're watching that is highly resistant to antibiotic treatment. It would grow exactly like Candida albicans does. So when you set up Candida albicans, you're going to be seeing what aureus would look like. But this is the safe version. This is the one that we can work with. So we're going to set up Candida albicans, which also, in case you're interested, is a yeast that you can actually buy to put in food as well. It gives different food flavoring. Candida tropicalis, which is an organism that can be associated with lung or skin infections in immunocompromised individuals. Again, we're just wearing our gloves because you'd have to have an open wound or something for the organism to get inside. And Candida krusei, which is actually kind of a control. This one rarely causes any kind of infection at all, even if you have a wound and pour it in there. All right, so again, don't try that. All right, just do your normal precautions. Okay, so this is a triplicate plate. Why am I not putting bacteria on here? Would they grow?
No.
No, because it's selective. Remember, we have a low pH, so it's selective. So we're going to set these 3 up, and then we're going to incubate them and see what they do. Word of caution. Make sure you are inoculating the correct plate. If I put the Candida on the bacterial plate, they will grow, but you won't see any reactions because they're not going to eat the sugars that are there. If I put the bacteria on the yeast plate, they're not going to grow. So make sure that you're reading the labels that are on the plates. This one says Hardichrome Candida. So that means I'm going to put the yeast on here, the Candida strain. This one says Hardichrome UTI. Hardichrome UTI. So that's my urinary tract. That's my bacteria.